Hey everyone, my name is David. Welcome to my poster. Here we're using population scale HLA typing to reveal the dynamics of T-cell evasion risk in individuals. And so in this work, we're really interested in studying the evolution of SARS-CoV-2 as well as its impact on T-cell immunity. And so a lot of our work focuses on HLA molecules. And so these molecules are found on the surface of virtually all cells in your body. And when a cell becomes infected by COVID, HLA molecules will actually present small fragments of viruses called epitopes to T cells. And so one thing that's really important to know about these molecules is that there's many different versions of the HLA gene around across the human population, meaning that we all have slightly different abilities to use our T cells to defend ourselves against the virus. And so while T-cell immunity was shown to be crucial during SARS-CoV-2 infections, certain strains, such as Omicron, were shown to lead to T-cell evasion in certain individuals, but not everyone. And so here, we hypothesize that the diversity in HLA alleles in a human population can actually explain variations in T-cell effectiveness against both existing and emerging SARS-CoV-2 variants. And so to address this, we aim first of all, to develop a computational framework to track the impact of SARS-CoV-2 evolution on T-cell evasion across many HLA profiles. And our second aim is to validate our findings in the lab using ELISpots. And so our work is done in collaboration with the Recover2 Biobank, which is a biobank of 576 COVID-19 convalescent individuals. And so for our work, it's really important for us to have the HLA types of all the individuals we're working with. And so to this end, we HLA typed our entire cohort using a novel high-throughput population-scale HLA typing method developed by our collaborator, Martin Smith. And so with this, we were able to HLA type all the individuals in our cohort. And as you can see, every individual has 6 HLA alleles. So HLA A, B, and C. And so with the HLA types acquired, we then proceeded to do 2 different things. The first one was to group or cluster together the different individuals of our cohort into different groups. And so, and our second aim was to predict the impact of SARS-CoV-2 variants on epitope presentation by HLA for the different individuals of our cohort. And so we did this using NanomHCpan, which is a machine learning based tool to predict epitope presentation by HLA. And we did this also using a comprehensive, exhaustive list of experimentally validated T-cell epitopes from the literature. And so this leads us to our first result figure, where we can observe the predicted T-cell evasion across the biobank. And although we did this analysis for many variants, here we're showing the results in the context of Omicron. And so in this figure, every dot corresponds to a unique HLA profile from our cohort. And the distance between the dots corresponds to how similar their HLA profiles are. And as you can see, we were able to cluster our cohort into 6 different clusters here, each cluster defined by its own particular set of HLA alleles, as seen in the boxes here. And so the color scheme here actually corresponds to the total number of T-cell epitopes that were lost due to Omicron mutations. And so darker dots lose more epitopes. And one of the key takeaways here is that T-cell evasion isn't the same across the entire biobank. We actually have one cluster here, cluster 1, that appears to be more susceptible to T-cell evasion. And this cluster is actually enriched in HLAs B0702, A0301, and A0201. And so, of course, this is something we see visually, but when we actually do the statistics using a 2-key test, we can see indeed that cluster 1, is actually more susceptible to T-cell evasion. And so what we see in the table here are the actual epitopes as well as the corresponding HLA alleles in both their mutated and unmutated forms for the 15 individuals that are most at risk of T-cell evasion who happen to be in cluster 1. And so what we can see here is we have enrichment in, of course, in alleles B0702, A0301, A0201, and A6801. And so this actually represents the experimental framework with which we're currently validating the loss of these epitopes amongst the individuals of cluster 1. And so finally, the last thing we wanted to do is we wanted to actually increase the statistical power of our analyses by replicating our work with a second, much larger biobank. And here we used the UK biobank, which is a cohort of 500,000 individuals, 488,000 of which are HLA typed. And so by replicating our analyses, we were again able to cluster the participants into different HLA defined groups. And again, we were able to identify one particular cluster which seems to be more susceptible to T-cell evasion. And once more, these individuals are also enriched in the very same HLAs that we saw in the Recover2 cohort, so HLA B0702, A0301, and A0201. And so in conclusion, we here developed a computational framework powered by high-throughput population scale HLA typing to identify human populations at risk of a reduced T-cell immunity to circulating and emerging SARS-CoV-2 variants. And this is relevant in the context of pandemic preparedness, as it enables the rapid HLA typing of large cohorts and the monitoring of T-cell evasion throughout the pandemic. And again, one of our main findings is that we saw that individuals enriched in HLA alleles B0702, B0301, and B0702, A0301, and A0201 were more susceptible to T-cell evasion by Omicron BA2.